guys want me to review anything from yesterday? Yes.
We were confused about the pathways for the erection, the sensory stimuli, and then the motor.
Okay. No, I just dim the light for that. Can someone hit the switch closest to the door? See if I can get that front light back on again. Yeah.
You're gonna need a really long stick. You're gonna need a really long stick so you can.
Yeah. I don't know why it's way over there. Okay, let's see if we can do this, if it works a little bit better on a sagittal view, like the first slide that I showed for the male. So I actually went to sleep thinking about how I could draw this better last night, so thank you for asking me. Okay.
It has better image stabilization.
And do you remember the colors? So sensory pathway for everything erection, emission, and ejaculation, all right, is all going to be through the pudendal nerve. There are some other sensory nerve fibers that are going to travel sometimes with autonomic components, but we're not going to worry about that part of it. All right. So absolutely change the color of sensory to the color of our neurons. You can recognize that. Remember, sensory is returning to the central nervous system. So our peripheral nerve is carrying afferent fibers, sensory information, back to the spinal cord. And this sensory information that we're looking at is coming from the glans penis primarily, because that's the highest concentration of receptors, okay, but anywhere along the shaft. And that's a component of the peripheral nervous system. It's the skin. So most of our peripheral nerves are combined sensory and motor structures, unlike the cranial nerves, in which we had primarily sensory, like the optic or the olfactory neurons, and we had primary motors like ocular motor or trigeminal. Okay, so this sensory neuron, or multiple neurons, are going to enter into the spinal nerve and pass in through the dorsal root. Their cell bodies are going to form the dorsal root ganglia. We're calling this from file 430. And then they will enter into the posterior gray horn. Okay. So the fibers that come together out of the penis and even out of the scrotum and out of other genitalia are identified as the pudendal nerve. at the point where they are in the penis, they are identified as the dorsal nerve of the penis. Okay. Following along with me so far? Now that forms a reflex arc when we talk about ejaculation, or rather erection. That forms a reflex arc with the autonomic motor neuron, which is not in the anterior gray horn, but we don't really have lateral gray horns, so it's kind of in between anterior gray horn and posterior gray horn. That's motor. It's autonomic motor, right? So it is going to come out of the spinal cord. Remember, this is S2, S3, S4 level. So we are parasympathetic. And will travel into the pelvis. By coming off of the ventral ramus and forming a bundle of neurons identified as the pelvic lengthening nerve. This is parasympathetic, the long preganglionic neuron that forms short preganglionic neurons that end up on the blood vessels of our erectile tissue, primarily corpus cavernosum, and some in the corpus spongiosum. Okay. So this later becomes the cavernous nerve. And these are motor, remember, so they're autonomic motor. And the neurotransmitter between the pre and the post is acetylcholine. And in the case at the penis is going within the arterioles, is going to be both acetylcholine and nitric oxide. Our neurotransmitters. Get back in bed. And this causes calcium to be returned to the sarcoplasmic reticulum inside the smooth muscle cells of the blood vessels. And decrease in calcium results in relaxation of those smooth muscles. Okay. And that's our arc. This is what I was talking about would occur even with the transection of the spinal cord, because we have this connection here. Now, if the person was not damaged in the spinal cord, this sensory would go up to the brain and would, I'm going to take a picture too so I can remember the group, would go up to the brain for actual processing and perception of that sensory aspect.
Post-ganglionic, it releases both acetylcholine and nitric oxide?
Yes. And the acetylcholine binds to those endothelial cells lining the blood vessels and causes them to release nitric oxide. Now I lost my screen. Okay, so that is erection. Now let's compare that to emission. Okay. that image from Netters. I just pulled it up now, we're gonna do. So if you call this little light, because we still have our lights out, but here coming out of the visual ray line, or the pelvic splanchnic, okay, that's what I'm drawing right here. So they don't have anything to do with sympathetic change. They're going to come out the ventral rami. And these ventral rami are, well, we'll deal with that when we get to ejaculation. But these are the ventral rami that were forming those. All of these are ventral rami, bundles of both motor and sensory neurons associated with the various levels of the sacral spinal cord. They're on the inside of the pelvic wall, actually on the surface of the obfuscator internus. Okay, so are we ready to move on to emission? Okay. So emission is going to be movement of the sperm from the vas deferens to the urethra, and secretions of seminal fluid and prostatic fluid to the urethra, and secretion of bulbourethral glands, Cowper's glands, through the urethra to the surface of the glans penis. So that's the only thing in emission that actually passes through the urethra, because remember it has to flush out the secretions ahead of time. So emission is going to involve a sympathetic innervation of vas deferens smooth muscle and the glands, glandular secretion of the seminal vesicle, prostate gland, and the bulbourethral gland, or Cowper's gland. Okay? So we're going to keep our sensory stimuli the same. So we'll keep the sensory neuron of the pudendal nerve there. With your permission now, I erase the parasympathetic because that will just add to our complication of our diagram here. And so we have our sensory stimuli coming in, and now this is sympathetic. So this information has to go up the spinal cord to the level where we have our sympathetic preganglionic cells, and that's up here in L1, L2. Okay? So it has to go up that high, and we have our lateral gray horns now. So we're going to have a crossing over. And this side coming in will also cross over. And so some of these neurons are going to end up in the sensory neuron level at the posterior gray horn at the L1, L2 level. Remember, they're going up anyway to go up to the brain for processing so you can perceive the sensation. So now our motor neuron is not going to be sympathetic, it's going to be parasympathetic. Sorry, other way around. It's not going to be parasympathetic, it's going to be sympathetic. It's motor, so it's still going to pass into the ventral root of our spinal nerve and into the ventral rami and come in through the ramus communicantes and descend down the sympathetic chain, along the sympathetic chain as it enters the pelvis. Okay, because remember our ventral rami from the sacrospinal cord have no sympathetic neurons. So the only way to get down into the pelvis is through this sympathetic chain. Now, because it is going to the organ, it's not going to synapse in the ganglion. This little connection here is for postganglionic neurons going out the sacroventral rami and going down your lower limb to get to sweat glands and blood vessels in your lower limb. Okay, so that's what this is for. So we're not using that because we're not worried about the lower limbs. This neuron is going to pass through the ganglion without synapsing and come into the pelvis. And so this nerve coming into the pelvis is a sacral ligament nerve. Within the rat's nest, the inferior hypogastric plexus, this is going to synapse with the postganglionic neuron. And then remember, it's going to those structures. All right, so it's going to go to, my positioning here is a little off. We'll have our testes down here, our epididymis, our vas deferens, going to the bladder, and then we'll have our seminal vesicles and our prostate gland. And yes, I recognize the prostate gland should be up here on top of the penis, but thematically I can't quite. Well, let me see, maybe I will allow me to move the penis down a little bit farther. Sorry, we just did plastic surgery on this guy. Okay, so we still have pudendal nerve carrying sensory information. This is going through, the reason I redrew that is because it's coming off here and then getting into the pudendal nerve and kind of coming in from the inferior aspect there. Sending information up to the lumbar level. Activating, this is going to happen on both sides, obviously. Activating preganglionic sympathetic neurons, which travel down the sympathetic chain into the pelvic cavity, pass out the ganglia without synapsing, and within the inferior hypogastric plexus synapse with their postganglionic neurons, which then go to the seminal vesicles, go to the prostate gland, and go to the bulbourethral gland.
Can you label those
Can we
structures?
put numbers on them rather than write the words out here? So, let's see. I didn't get vas deferens in here. So, 1, 2, 3, 4. Seminal vesicles 1. Prostate gland is 2. Bulbourethral gland is 3, and vas deferens is 4. So sensory stimuli travels to the sacrospinal cord, travels up to the lumbar level spinal cord. Motor innervation comes out. We have acetylcholine here, between the pre and the post as always, and then norepinephrine at the receptor sites within the glands and the muscles.
What was the vas deferens?
Yes.
What was-
Smooth muscle of the vas deferens. Okay? So now our last step is ejaculation. So we use all three of our different components of motor innervation. I probably should have put sympathetic as a different color because I'm going to leave that on there and add parasympathetic back. Yes.
And so could you have it, if I'm taking it, the preganglionic fiber of the motor pathway is going through the white communicative? Is that what that's showing? Okay.
And then this one and this one would both be gray.
Yeah.
You're allowing postganglionic to get back to the ventral brain line.
Let's see.
Okay? Any questions on this before we move on? All right, so now ejaculation. We're still continuing the same sensory, okay? And but we're still going to see our pudendal nerve. Let me just kind of write that up over here. Sympathetic innervation now is going to go to the internal sphincter of the urinary bladder. So we'll erase this. Not because it's not there, we're just not trying to emphasize this at that point. The internal sphincter is between the bladder and the prostate gland. And then we have our urethra. It's going to travel down through the penis. And then we have our ischiocavernosus muscle and our bulbospongiosus muscle. As you can see on this screen, as here and here. I have a sagittal view up here, so let's go back to a sagittal view. So what we're talking about with the three different innervations in this sequence is the internal sphincter of the urinary bladder, the smooth muscle of the urethra, right, and the skeletal muscle of the bulbospongiosus and the ischiocavernosus, which are off the screen, because they're in front or behind of this image. So sensory is still pudendal nerve, and sympathetic innervation, still coming through this pathway, is going to go to the internal urethral sphincter. And I will draw parasympathetic as a red dotted line since I'd used red previously. So parasympathetic is still going to be from here. It's going to come through the pelvic splanchnic nerve just like it did before. It's not going to come via the cavernous nerves because the cavernous nerves went to the corpora cavernosa. All right, so now we're just going to get to the smooth muscle of the urethra. Sacral splanchnics, pelvic splanchnics are doing urethra, sacral splanchnics are doing internal urethral sphincter. There's actually both of them are involved, but remember parasympathetic is primarily the activation of our urinary system, so I'm just focusing on that. And then finally we need voluntary motor. And that's going to be doing bulbospongiosis. And ischiocavernosus. So this nerve is sacral splanchnic. This nerve is pelvic splanchnic. And what would this nerve be? Pudendal. Alright, so let's do that one in blue. So this is going to be coming down from the spinal tract, right, from the cortical spinal tract. And it will have already crossed over in the medulla. So it's coming down the same side as our anterior great horn. It's going to come into the anterior great horn. That's our upper motor neuron, bringing that information down. And pudendal nerve is at the sacral level. So we're going to have our lower motor neuron coming out through the ventral ramus. Okay. Comes out a little bit farther than the pelvic splanchnic, and it's going to travel with the sensory neurons of our pudendal nerve. And it's going to come down and pass through the greater sciatic foramen and the lesser sciatic foramen and give fibers to our skeletal muscle. I said I was going to do this. Never mind, I'll leave it that way. I was going to do it dotted, but okay? all over that. And then if you have any questions. By the way, this pathway of innervation is exactly the same for the female. Right, so we have erectile tissue in the female that we'll talk about on Tuesday. Same pathway of innervation. We have emission, so we have some prostatic equivalent glands that are around the urethra. And we also have ejaculation of that emitted fluid. Now, every female experiences that, but in 2001, these glands that you see here on either side of the urethra were scientifically agreed upon to be equivalent to the prostate glands. So some women talk about having an ejaculate, and everybody would tell them for years, no, that was just some urine, well, it wasn't acidic. And so that's been officially recognized as, now whether women can get prostatic cancer through this, you don't hear about those glands becoming tumors, but they are considered now to be functionally the equivalent. Okay, may I erase the board? No one said yes, someone said no.
I'm pretty sure I did, yeah.
It's too late now.
No, I'm going to double check. Yep.
We're halfway through lecture time. That's okay, this hard stuff is hard to understand. Female anatomy is a little bit better. Do you mind hitting that switch again? Let's see if that one switch does what I need. Okay. So the female reproductive system, the easiest contrast to see right off the bat is obviously there's no penis. And there's no scrotum. Okay, so our gonads are maintained within the pelvic cavity. But as we go through this, we'll talk about some of those equivalent structures. So remember the mesonephric duct was what formed our epididymis and vas deferens, the top part of the urethra. That disappears in the female, and instead the paramesonephric duct remains. And that is going to form the oviduct and the uterus, and about this much, the top 20 to 25% or so of the vagina. There was another film of female form. I put that link up already. So again, pronephros and metanephric duct. Mesonephros starts to appear in the female as well. That induces the development of the ovary. So now we have the mesonephros and the mesonephric duct. We're starting to see the development of the ureters and the metanephric kidney, which is the functional kidney as well. So all of this is still the same in the female. We have the appearance of the gonad induced by the mesonephros. And now you're starting to see that blue structure, which is the paramesonephric duct. The mesonephros disappears. Those structures that connected the testis to the mesonephric duct, the efferent ductules, actually remnants of them remain in the female, but they're connected to the disappearing and absent mesonephric duct, and they do not connect the ovary to the paramesonephric duct. So while we can still see some remnants, there is no specific connection here between the ovary and the oviduct. Hence, the released egg is free to move anywhere, not just into the oviduct. The two paramesonephric tubes fuse to form a common uterus. Some animals will have two uterine horns. So like in a cat, you can have the fetal developing kittens and two separate uterine horns. and then fuses to form the upper portion of the vagina. Okay, so let's watch that one more time. So do you guys remember what causes the disappearance of the mesonephric duct? So what are we missing in the female? We're missing the Y chromosome. And on the Y chromosome is an SRY gene. And so we're missing enzymes and chemicals produced by that cause the different disappearance of the mesonephric duct. That cause the disappearance of the paramesonephric duct. And we have molecules in the female that cause the disappearance of the mesonephric duct. So in the male, there's an anti-Müllerian or an anti-paramesonephric chemical that's released that inhibits and causes the paramesonephric duct to actually disappear. And that is synthesized by genes that are on the Y chromosome. So in the absence of the SRY gene, we keep the paramesonephric duct and lose the mesonephric duct.
So is this fusing why some women can be born with 2 uterus or 2 vaginas?
I had a patient once who had 2 vaginas. She came in to deliver her baby. She didn't have 2 uteri, so her OBGYN joked with her that she could have intercourse in one, and that could be her birth control, and use the other one for fertilization. But yeah.
How does that work? Like they're next to each other?
Yeah. The blind ending one was more anterior, so you had 2 vaginal openings. All right, so when we look then at the development here between comparing male and female, so what we just visualized, this is still the same, and as I indicated earlier, without the SRY gene, the mesonephric duct disappears, and in the folded peritoneum, between the gonad and the paramesonephric duct, we can actually still sometimes find the components, the mesonephric tubules that form the efferent ductules. In fact, we have a model, quite an old model, vintage model, if you will, in the lab that I'll show you on Tuesday that still shows those structures. All right, so those then eventually fuse and create the uterine tube, cervix, and this upper portion of the vagina. So a much simpler tubular structure, not as many different glands and structures to describe. We will make up for that when we start looking at the uterine cycle and hormonal cycle. So here's a difference in the description and the formation of the external genitalia. All right, so in the male, we have the fusion of this tissue here that creates the wall, the shaft of the penis, and the fusion of these folds that creates the scrotum. Remember I said if you have access to legal channels and moral channels to a male, take a look at this ventral surface of the scrotum, and you'll be able to see that seam line. Okay. So an infant, if it's a toddler, and you're not its parent, I would go look for a baby or something. But you can actually visualize that seam line. So in the female, without the expression of the SRY gene, remember we said that developing infant would take a female structure unless things change that. So here we have the two, we'll look at this on Tuesday in the external genitalia. And we have these 2 outer folds known as the large lips or the labia majora. So in women, those are the outer ones that have hair follicles on them, unless you've had a Brazilian or had laser or whatever. So those are the equivalent. Remember I talked about a list of equivalent structures, homologous. Homologous means equivalent to. Those are the structures that in the male will fuse to form the scrotum. So labia majora are homologous or equivalent to the scrotum. And the inner lips, which are thinner and do not have hair follicles on them, are called the labia minora. And they fuse to form the skin shaft around the penis. Okay. So there's surgical procedures for individuals who want to change their phenotype from male to female or female to male. And so you can find images of this online. When I taught reproductive physiology a couple years at Sex State, they had to do case studies. And so a group of them decided to present that information. It was quite a weird and amazing the things that they can do surgically to create a vagina. They actually use the skin of the penis and turn it inside out, or put it not inside out, but kind of push it up into the opening they create for the vagina. They don't make a uterus, obviously, and they just take the testes out, and the male to female starts taking female hormones. A little bit easier to go from male to female, remove the testes, open up the scrotum, reduce the penis to a clitoris, and still maintain nerve endings. Now, occasionally, a male to a female decides they want to go back again. That's a little bit more difficult, okay? Because a female to male is a little more difficult. You can create with transplants, and then there's also penis pumps that were developed for men who had had prostate surgery and damage to the dorsal nerve, so they can pump fluid and create an enlarged penis. So that type of a component can be done. They don't really get all the actual rectal tissue of an actual penis. So there's quite a large number of advances that have been made in the last 30 years in that type of surgery.
Have they started to do, have you ever done transplants, like from a cadet or no?
Of penises or?
Person? Yeah, like for the
I know they tried to reattach
yeah.
Penises, and certainly microsurgery has become much more successful. I haven't read about it, but I wouldn't say it hasn't occurred.
They've done it with the whole face.
Right, the whole face and stuff. So they certainly can reattach nerves. Sometimes all the feeling isn't there. I'd have to check on that. I haven't read about that happening. And when this case study was done a couple years ago, they did mention it. All right, so looking at we're going to look at the changes in the ovarian cycle on Tuesday. But the ovaries are obviously the female gonads. They have a capsule on them known as the tunica albuginea, similar to what we had in the male. They also have a germinal, it's called a germinal epithelium. I'm just talk about it here. A lot of times, for a while, people thought that this is the origin of the oocytes. It's just a simple cuboidal epithelium. It's not germinal in any nature. Okay? So when you see the word germinal, it doesn't provide any germ cells, which would be the oogonia. The ovary is suspended within the pelvis by ligaments. And remember I diagrammed the testis, and we had our male gonad, and it had an upper gubernaculum and a lower gubernaculum that ended up in the scrotum. Right? So in the female, we have equivalent or homologous structures. So in the male, this is upper gubernaculum, sometimes known as a suspensory ligament. In the female, this is identified as, it's not showing up well. This is identified as the ovarian ligament, or ovarian ligament proper. There is a suspensory ligament in the attached to the ovary, but I don't want you to confuse it with the suspensory ligament overlap of the male name. So there are two ligaments attached at either of the long poles of the ovaries. So this doesn't work. Up here, where the retractor is, you'll see a red and blue. Those are ovarian artery and veins associated with the abdominal aorta and inferior vena cava. And then that suspensory ligament you see is a cord of tissue that goes up to the posterior abdominal wall as well. However, the ovarian ligament that you see here, okay, is part of that upper gubernaculum. And then the rest of it, what we see down here as the gubernaculum of the in the male, the lower gubernaculum. In the female, is actually attached to the uterus and is known as the round ligament. Remember what I said just a few minutes ago? The scrotum in the male doesn't fuse and is seen as what structure in the female? The labia majora. All right. So in the female, then, the round ligament of the uterus is attached to the labia majora, and it's the only structure keeping the uterus tilted forward. So if we go back to this diagram, here's the uterus. You can barely see there's a round ligament here passing through the deep inguinal ring, just like the vas deferens and all the other structures would have passed through the deep inguinal ring and gives access to the labia majora. So the ovary is supported then by the suspensory ligament to the abdominal wall and the ovarian ligament proper to the uterus. It is partially enclosed by peritoneum. Peritoneum is that simple squamous membrane, serous membrane that lines the abdominal cavity. If it was completely enclosed by it, what would our problem be?
Nothing to get out?
Yeah, the oocyte would be trapped under that membrane and would have difficulty getting access to the oviduct, which is underneath it, but if it's completely surrounded by it, it would be like wrapping it in shrink wrap. Now, we'll come back to this image on Tuesday. I just want to give you a heads up. All these small circles that you see here are the developing oocytes. As they move to ovulation, the cells around them start to multiply, why we get increasing levels of hormones. This is an artistic illustration of the changes that those follicles go through. But in actuality, this oocyte right here is going to stay right here as it goes through all these changes and be ovulated from that point. So it doesn't migrate around the ovary and get ovulated from a different side. This was just, the artist couldn't show all those changes on top of each other, so it put them in different locations. But all of those changes in the development of those follicular cells would occur at each point where the oocyte is located. The oocytes are found along this outer portion of the ovary, along the cortex. The medulla or inner region is where the blood vessels, either from the ovarian artery and vein or from the uterine artery and vein, will come in. This ovary is flipped from what we're looking at, so the uterus is over here. This is our ovarian ligament proper. So we'll come back and talk about all those stages on Tuesday. Now let's follow the released oocyte, and it's going to move into the uterine tube. So there are several different terms for uterine tube. I'm on page 92. So there is uterine tube, there is oviduct, there's an older name which is fallopian tube, and there's the Latin word, which is salpinx. Okay. Probably oviduct and uterine tube are the most commonly used. And salpinx, the reason I mentioned salpinx is the surgical, a woman will talk about getting my tubes tied. It's also known as a tubal ligation. The surgery itself is a salpingectomy. Okay, ectomy is cutting. So cutting out the salpinx is the same as a tubal ligation, or the equivalent of a vasectomy. So ectomy is that term, cutting. Okay. Now, if you look, there are several components, several regions to the structure of the oviduct. The ovary is here. The oviduct and the uterus is over here. The oviduct, at the point where it's most closest to the ovary, has long, motile, finger-like structures that look almost like the anemone in the ocean. And these are identified as fimbra, or fimbri, plural. And they can move. All right. So at ovulation, the whole oviduct actually comes closer to the ovary and actually touches it, and these fimbri of the oviduct move to create a current that sweeps up the released egg and surrounding cells. Then this tapers kind of like a trumpet-like structure and narrows. So this widest portion is identified as the infundibulum. And then most of the oviduct itself is identified as the ampulla. And this is where fertilization typically takes place. If fertilization of the egg takes place out here, or in the infundibulum, it has a higher probability of implanting within the oviduct itself, which might last for a few days, but doesn't have the capability of stretching. And if the tissue continues to live, if the embryo is implanted and developing, the oviduct can rupture. And then subsequent scar tissue often blocks that uterine tube from being available for the released egg to pass through. The oviduct narrows as it passes through the uterine wall. And so at that point, it's identified as the isthmus. So 4 regions of the oviduct. So this is a part of view of the oviduct actually as it's closer to the uterine tube. Let me pull up the image we had yesterday. The epithelium that lines the oviduct is simple columnar with cilia. And those cilia, let me draw this higher where you guys can actually see it. Are motile, and they sweep the oocyte towards the uterine cavity. So the oocyte is going to be going in this direction, and obviously the sperm are going to be going in that direction. When the sperm swim and are propelled up the uterine cavity, they actually form a pool just outside the opening of the oviduct, and then they will move several times as about 100,000 at a time. They will move into the oviduct. So they don't all enter the oviduct at one time. with about several 100,000 or so at a time, they will move into the oviduct. Now the ovaries release typically an oocyte from one ovary, and then the next month from the other ovary, and so on. And the sperm seem to be able to detect which ovary is going to release the oocyte or has released the oocyte. And they will have a higher number clustered at that particular oviduct. So it's thought probably to be due to the fact that they can sense release of hormones and chemicals from the egg that is that developing ovary, oocyte or released oocyte. So if the oocyte is being moved along through this area in here, right, the sperm are actually down in here. And they are moved by peristalsis. So that rhythmic contraction that we talked about, moving the sperm through the vas deferens and the urethra, and our food through the digestive tract, that same type of rhythmic pushing is moving the sperm, and they're going to be down in this region while the egg is up. That's how they can go in opposite directions. Okay? Or at least until they meet. So now that brings us to the structure of the uterus. And in many of the diagrams of the uterus show it looking like this fashion, like an upside down pear. And it makes it easy to draw the structures of the uterus. In actuality, the uterus is bent at right angles at its cervix portion here that narrows. And so it really, in a sagittal view, is tilted forward over the bladder. Right, so here's the urinary bladder, here's the uterus, and then its cervix is typically bent at an angle from that. That's called anteflexion. I have those terms on the next page in your lecture notes. Any degree that's more posterior, more straight, or more posterior is identified as retroflexion. So even though when we look at the uterus kind of upright, all arms stretched out like this, so pretend I'm a uterus, right? So this would be the top of the fundus, and the body, and the cervix. Bladder right here. So the uterus is bent over like that, and then the oviduct, instead of being out straight, is actually posterior and lateral. So the ovaries and the oviducts are swept back to the side and behind the uterus itself. The top part of the uterus is called the fundus. We call the top rounded portion of the stomach the fundus, the top rounded portion of the bladder the fundus. Okay? That's this region right here. So if you're pregnant and they're measuring how the uterus has developed to determine how far along you are in your pregnancy, they'll put the top of the tape measure up where they can feel the fundus, down to the pubic symphysis. Then we have the body, which is a major component, and then this narrowed region is the cervix. And this would be the uterine cavity. So implantation, proper implantation, is going to take place in the uterine cavity, and typically along the posterior wall, the surface of it, more dorsal aspect. This channel in the cervix, some of you can't see that, so I'll redraw it over here in just a moment, is known as the cervical canal. And the opening from within is known as the internal os. And the opening from or into the vagina is known as the external os. I'm going to erase this part of the femur and I'll just redraw the cervix up there for individuals.
So the external os down at the bottom.
The external os would be what the sperm would pass through first to enter into the cervical canal. So this would be the external os right here. So if a woman uses a diaphragm, the diaphragm is fitted around the inferior aspect of the cervix and covers the external os. That's the first thing the sperm are going to pass through to enter into the cervix. Okay. So I'll redraw that aspect up here because we need to look at it anyway from the vagina. Epithelium of the uterus is simple columnar as well, without the cilia. Okay, so this would be the cervical canal. Someone goes, has an annual pelvic exam, and they do a pap smear. It's named after a Greek physician, Papinicolau. I don't know if I'm pronouncing it right. That's why they shortened it to pap. And that's a scraping of the epithelium lining the cervix, and they look for various changes in those cells, and they categorize them in various stages depending on how much change has occurred. So again, this would be the internal os, and this would be the external os. Now the uterus has a lining of epithelium. We'll get to that in just a moment. It is formed of smooth muscle. That is the myometrium. So if you recall from Myo 430, myo was one of our roots for muscle, myo and sarco, and metrium means uterus. All right, so the smooth muscle of the uterus is the myometrium. This is what has receptors for the oxytocin from the posterior pituitary that results in contraction. There can be smooth muscle tumors that are benign, known as fibroids. Okay? So tumors of these structures, this muscle tissue, are known as fibroid tumors. While they are not malignant, they often can result in heavy bleeding during and in between a woman's period. They can be very small to grapefruit size. About 6 or 7 years ago, one of my students actually started bleeding during our exam on the reproductive system. Went home, lost so much blood that her hemoglobin was about a 4 instead of a 12 to 14. And she knew she had fibroids, but had been trying to deal with it without surgery. But they removed that grapefruit size type of fibroid tumor from her. They can come back, so just removing, if you've got lots of small ones, to go in there and take them out surgically may not be the proper treatment at that time because they can return. All right, the covering of the uterus is a continuation of the peritoneum, and that's only on part of the uterus. So here you can see that serous membrane. Again, the abdominal cavity is lined with peritoneum. And it continues, let me put up the sagittal view, that might be a little easier to see. So here's a sagittal view. The membrane here is a simple squamous epithelium attached via loose, irregular connective tissue. That's what a serous membrane is. And it's the parietal peritoneum when it's attached to the abdominal wall. Remember we talked about parietal pleura and parietal pericardium and visceral structures. So here it covers the top of the bladder. If you have a really full bladder and it moves up into the abdominal cavity, more of it gets covered with peritoneum. Then it dips down and forms a fold back onto the uterus and comes down about to the cervix in the back, not quite as low in the front, but part of the cervix in the back, and then it folds back up again to go onto the rectum and posterior abdominal wall. So where it is attached to the uterus, it is identified as not the peritoneum, but the perimetrium. Okay? So we have, back to this real simplistic diagram of the uterus then, our smooth muscle, would be myometrium. The peritoneal structure is perimetrium. And then we have a layer of epithelium and connective tissue lining the uterus. So the blue line would be the epithelium, simple columnar epithelium. And this is endometrium. So you may have heard of endometriosis. Okay, that's the endometrium. Portion of it is what separates from the uterine wall and forms a woman's menses with each monthly cycle. When a baby is born, that's the afterbirth. All right, so the placenta, tissue, and all the surrounding upper portion of the endometrium has to separate and leave the inner cavity of the uterine cavity. Now, the few minutes we have back, let me just introduce you to some of the supportive structures or ligaments of the uterus. So these are diagrammed on page 94 as well as on page 96. Now this is the view that I first drew of the uterus, kind of looking like this without showing the indication of the bend. All right, and it makes it a little bit easier to understand the relationship of the peritoneum and its structures than the cross-section, the sagittal view. So again, I'm the uterus, all right, and if I take a sheet and drape it over me so that it's hanging off, kind of like if my arms or upper limbs are clothesline, it's hanging down from either upper limb. Where that a sheet is attached to the top of me and to my body, that would be the perimetrium. That's what we're seeing here as that shiny cover. All right, so this is perimetrium. However, where it's hanging down from my arms, it forms a double layer of peritoneum. All right, I don't want to stretch my sweater out too much, but if we consider this to be the peritoneum, you can see that where it's loose and hanging down from my arm, there's two layers. Simple squamous on either side, loose connective tissue in the center. When we get to the digestive system, we'll learn that any time we have a double layer of peritoneum, it's called a mesentery. Okay, we see a mesentery between the stomach and the liver and several other organs. So what was our root word for metrium? I'm sorry, for uterus? Metrium. All right, so the mesentery of the uterus is called a mesometrium. That's what we refer to this layer portion that's right here. However, we have a double layer coming down from the oviduct and a double layer partially enclosing the ovary. And that can be seen like this in cross-section. Can I erase the board here? And we'll just, and I want to kind of re-diagram this, okay? So this is, the uterus is behind the board, all right? This diagram has been made by making a cut, where am I going here? Making a cut through here. Actually, this is probably an easier one to understand. So here's the uterus. Here's the peritoneum, all right? And this would be the mesometrium, which is associated with the body of the uterus. So as we look at the oviduct, all right, so here's our oviduct. Over here is our ovary. What color was my perimetrium in the first diagram? Green. Green, all right. So this sheet that's hanging over my body, as it hangs over the oviduct, comes down now and forms a double layer. we take part of that sheet and pull it out and use it to partially suspend the ovary. Remember, we don't want to go all the way around it or we completely enclose those oocytes. We just go partially around it. Then the rest of it comes back down and it's going to go up forward over the bladder and then back over the rectum. But this is our mesometrium structure. Now technically, instead of calling the whole thing the mesometrium, the term broad ligament is used for the whole thing. And this is the mesentery of the oviduct, known as the mesosalpinx. Mesentery of the ovary, known as the mesoovarium. Mesentery of the uterus, known as the mesometrium. About a minute over, so we'll stop there and come back and talk about the vagina, external genitalia, elaborate on sometime on Monday, and then we'll also start the hormones and ovarian cycle and so on. Not Monday, sorry, Tuesday. So we finished up female reproductive next week. Tuesday, you need to bring the modeling clay or Play-Doh for modeling the rectal tissue. All right. Thursday, we'll talk about fertilization. And we have, if the sea urchins are happy, we'll actually fertilize sea urchin eggs. And watch the little sperm swim around and attack those eggs. They should be coming in the mail on Sunday or Monday. And then we get them to release their eggs with injections of potassium chloride. And then on Friday, we'll talk about implantation. All right, what happens after the egg is fertilized. Tuesday, I didn't want to do an exam right after that because I know this is a lot of complicated mechanisms that we've been covering. So the following Tuesday, if there's any material I need to catch up on, or set as a review day and our case study presentation. Because I know we've covered a lot and just slammed me with an exam that Tuesday without giving out more opportunity to get that a handle on it. I didn't think it was really fair this first unit. So I added in an extra day there, and I needed to do that also with the sea urchin schedule the sea urchin lab. So it's kind of our schedule, but the new content should be done with by the end of next week.
Yeah.
Have a great weekend.